First of all, the motile fungi, as the name indicates, these are the fungi which has at some stage they have some motile partners, motile things in their life cycle. For example, some spores, for example, presence of zoospores that have some kind of motility organs like flagella. So many members of this fungi have already been removed from being true fungi, right? I mean, they are no more considered as true fungi. They are considered as pseudo fungi. For example, true slime molds. It's a group that's known as that's also known as Zygomycetes. The Zygomycetes or true slime molds are those fungi that don't have their cell walls, so their protoplasts are visible with naked eyes. This group has a big diversity of organisms, from being microscopic to macroscopic. There's a very important species that is used that is used as an experimental model. For example, the Phycomycetes. So these members have flagellated cells in their life cycle. For example, they have swarm cells. These swarm cells can swim in presence of water because of presence of flagella. Then they feed on smaller organisms like smaller spores or bacteria. They show engulfing behavior. So as I discussed earlier, the role of predator. So here you can see these are playing important role in the soil ecosystem as a predator by engulfing the smaller organisms. So then they are also important saprotrophs. Another group of motile organisms are known as Oomycetes. Oomycetes include some important plant pathogens such as Pythium and Phytophthora species. Then another important groups, for example, catrids. Catrids are the true fungi and hyphocatrids. Actually, if I divide all these groups like true slime molds, water molds, and hyphocatrids, I can place these three groups separately. That is, true slime molds, water molds, and hyphocatrids can be placed separately in a separate group. That is, we can call it as pseudo fungi. So they have already been removed from true fungi. However, only the catrids have been placed in true fungi. These catrids are basically parasites of different animals and algae, and there are some saprotrophs which are also present. These saprotrophs use organic matter for their nutrition, especially they can dissolve chitin and cellulose and pollens. Then important group of true fungi is Zygomycota. Zygomycota is a very is a group that contains a huge diversity of fungi. There are remarkable differences in phenotypes, lifestyles, and nutrition in this group of fungi. They have aseptate hyphae. They lack septa. Then some fungi are symbiotic or host dependent, and such fungi cannot be cultured without host. Others are saprophytic. They can use the dead organic matter for the nutrition. Then there are plant symbiotic fungi such as mycorrhiza, especially arbuscular endomycorrhiza. They come from the fungal member of arbuscular endomycorrhiza belongs to glomales. And glomales are mostly placed along with Zygomycota, although sometimes it's placed separately than Zygomycota. Then there are some animal symbionts like Trachomycetes. They are symbiotic in guts of insects and nematodes. And then there are Zygomycetes, which can use or proliferates on breads, such as bread molds. And then there are many microparasites which use other fungi as food. Then the next important group of fungi are Ascomycetes, yeast and mold. They are also known as sac fungi or cup-shaped fungi. They are known as sac fungi because of sac-like structures they contain. These sac-like structures are present in the fruiting bodies. They contain the sexual spores known as ascospores. These sac-like structures are also known as ascus. As this figure, it's clear that each ascus contains a number of spores. These spores are sexual spores, and they are also known as ascospores. You can see different numbers of spores. For example, in this case, there are 8 spores in each, and there are different number. So this number vary from 4 to 8. So up to 8 ascospores are present in each sac in case of yeast, in case of Ascomycetes. So in this case, this group of fungi is an important member of higher fungi. The mycelia are septate, although these septa contains pores through which the mycelia can move around. So mycelia along with nuclei that can transfer from one part of mycelium to another part. So another thing that I would like to discuss in this slide is the the relationship of Ascomycetes with Deuteromycetes. Deuteromycetes basically contain the asexual forms of fungi. The asexual forms are also known as the anamorphs. So as the soil microbiologists mostly deal with the shapes of fungi which are present in soil, and mostly the fungi present in soil are present in their asexual forms, and these are basically anamorphs. So either their sexual forms are known or not known. So two things can be possible. In some cases, the sexual forms are also known, and if the sexual forms are known, they are separately named, but they are basically teleomorphs. So when we talk about Ascomycetes, we are talking about the teleomorphs sometime, because when we talk about Ascomycetes, we are talking about ascospores, and ascospores are the characteristics spores of Ascomycetes, and ascospores are the sexual spores. So they are present in the sexual forms of the fungi, and those sexual forms are basically known as teleomorphs. But these sexual forms may have their counterparts, their asexual form, that's called anamorphs. And anamorphs are basically the asexual form of the same fungi. These asexual forms have been placed in Deuteromycetes. So here we conclude with this statement that a fungus may be genetically same but may have different phenotypic identification based on sexual or asexual forms. For instance, I would like to give you two examples. One is the Trichoderma. Trichoderma is basically a soil-borne fungus, and Hypochondria is basically another fungus. So these are two forms of the same, genetically same fungus. Hypochondria and Trichoderma are genetically same but morphologically different. This is what I want to tell you. Now, Hypochondria is basically the sexual form of Trichoderma. It contains ascospores. Trichoderma doesn't contain ascospores. It contains asexual spores known as conidia. Now, the two types of spores are different because one are produced not by combining nuclei at any stage. So there are no combination of nuclei. That's why some spores are known as asexual spores. For example, the conidia. Trichoderma have conidia, while its counterpart, its sexual counterpart, that's known as Hypochondria, it contains sexual spores. Similarly, there is another example, Aspergillus and Eurotium. Now, Aspergillus is basically anamorphic form, and Eurotium is its sexual counterpart. So the two types of fungi are genetically same, but they are morphologically different. So Deuteromycetes contain the fungi for which sexual forms may or may not be known. This is very important to know, that all the members of Deuteromycetes are basically anamorphs. Some anamorphs have their teleomorphs known, but some may not be known till now. The majority of Deuteromycetes have their sexual forms in Ascomycetes. So this is very important to know, that Deuteromycetes have their sexual forms and majority have their sexual forms in Ascomycetes, as I have already given two examples. That's Trachoderma, and then another is uh, as here another is Aspergillus. So, however, we all of them are not Ascomycetes. Some belong to Basidiomycetes also. So we will be discussing Basidiomycetes later on in our coming slides. So as I already discussed that as soil microbiologists are dealing with soil, and soil are present, I mean, in natural environment, and mostly asexual forms are present in the soil. Sexual forms are found, I mean, a bit rarely by soil microbiologists. The most commonly found soil Ascomycetes fungi are Aspergillus species, Trachoderma species, Penicillium species, Fusarium species, Falciparum species. All these fungi are asexual forms, and they are anamorphs. Similarly, there are other members like Coletotrichum and many more examples. Now we, in this slide, we discuss Ascomycetes as pathogens. Generally, the fungi are opportunistic pathogens. Some of the well-known, I mean, notorious pathogens of plants are Coletotrichum species. They infect very big diversity of plants. Similarly, Fusarium species are very dangerous pathogens of plants. They infect all kinds of vegetation. They can cause disease in roots, leaves, and all kinds of disease. They are caused by Fusarium. Similarly, Alternaria species. Then there are many examples of pathogens. Then some well-known pathogens of human animals that belong to Ascomycetes are Candida species. Candida species are very common in human beings. They cause candidiosis. Then histoplasma species are there. They cause histoplasmosis. And then coccidioids and Aspergillus species. So they cause mycosis in humans and animals. They cause skin problems, allergies, and different kinds of problems and diseases in human and animals. Then the Ascomycetes are used in industry. For example, a number of Ascomycetes have been used to produce different types of antibiotics. Some examples are penicillium, produced by penicillin species. Penicillin produced by penicillin species. Then cyclosporine, that is produced by Tolypocladium species. And then loastatine, that's produced by Aspergillus species. And when we talk about industry, we can't forget yeasts. Yeasts are members of Ascomycetes. And example is brewer's yeast, that's very commonly used in production of bread. Then I would like to discuss some features of yeast here. Yeasts are basically unicellular fungi, and when they grow and then they reproduce by dividing the single cell into two. Now, either this division is by budding or by binary fission. Budding means that a small outgrowth develop on the cell, and this outgrowth starts growing, increasing in size, and then makes a new individual. That individual sometimes remains attached and make a kind of chain or group of cells together. Binary fission means that the cells divide into two,